Hi, my name is Lisardo Fernandez. My role in the Networks and Virtual Environments group is as researcher. In this presentation, I will review the use of parallel architectures in DNA methylation analysis. This review focuses on all the aspects of DNA methylation analysis that are alignment and methylation context, detection and visualization of differentially methylated regions, as well as the use of artificial intelligence techniques in methylation analysis. Finally, we will take some conclusions. DNA methylation is a representative mechanism of epigenetics. Epigenetics refer to changes in DNA that do not change the actual DNA sequence, meaning that these changes are potentially reversible. Shortly, DNA methylation consists of the addition of a methyl group to a cytosine, forming a 5'-MSE link. When this happens, that DNA position is methylated. DNA hydroxymethylation happens with an oxidation process over the methylated cytosine, and all of this process can be restored to a non-methylated cytosine. Methylation has been identified as one of the epigenetic processes which seems to play a relevant role in the genetic regulation because of its role in local control of gene expression. In this way, behind of each hand, there are a transcription region. If this region is hypermethylated, the gene expression is inactive. This feature is crucial in the cell behavior. It can cause genetic malfunction and, at least, diseases. DNA methylation analysis requires a specific treatment of DNA that modifies sequence as well as software tools of their analysis. The methylation data can be obtained through visual field sequencing, which provide comprehensive DNA methylation maps at single base pair resolution. The first step is the DNA extraction. Next, a polymerase chain reactions breaks up the DNA sequence into many fragments and makes multiple copies of these fragments, which are called reads, and are the actual samples to be analyzed. At this point, visual field is added to the resulting DNA reads. Visual field treatment converts unmethylated cytosines into thymines, while leaving methylated cytosines unchanged. Finally, the visual field reads are processed by a sequencer, which reading each read yields a text containing the sequence of nucleotides in this read, together with other meta-information like quality of the reads, etc. Since the sequencer carried out this process with all the reads, the output of the sequencer is a text file, is a FASTQ format containing the sequence detected for each read and is usually HU. Initially, the most important limitation was obtaining the sequenced files. It was really expensive in time and money. The next generation sequencers technology enables mass production at low cost. So, the genomic analysis efficiency depends on software tools with HPC solutions. Then, we'll review the different tools proposed for the different steps and aspects of the DNA methylation analysis, focusing on the methods they use for offering an efficient analysis to biomedical researchers and the use of the underlying computer architecture that each one is able to carry out. The first step in the DNA methylation analysis of a biological sample consists of aligning each read and detecting its methylation status. By aligning and comparing visual field sequencing reads to the reference genomic DNA sequence, it is possible not only to align the read, but also to infer DNA methylation pattern at base pair resolution. However, the data to be processed are huge, and a high computational power is required for an efficient operation. For example, the length of the DNA chain in the human genome is 3 billion nucleotides. There are two main techniques to align the reads from FASTQ files: wildcard algorithm and three-letter algorithm. Instead, to focus the attention in the underlying maths and tricks, let's see how it is implemented. From the point of view of HPC, we can see that there are shortage of tools implemented efficiently. Only RRBS map and HPG methyl exploit the degree of parallelism in current multi-core processors. In fact, the tools developed for heterogeneous architecture using GPUs are currently outdated, with the new length of reads in FASTQ files yielded by the current next generation sequencers. The tools discussed in the previous sections provide the user with single-base methylation results, indicating the absolute methylation level of each nucleotide found in the DNA of the analyzed sample. Most of them yield the results in large sequence alignment map (SAM) or binary alignment map (BAM) files. Thus, the analysis of these results is far beyond the human capabilities. Moreover, an effective biomedical analysis requires the identification of those areas of the genome where the methylation level of different samples significantly differ. These areas are called differentially methylated regions, DMRs. In order to detect a DMR, several BAM or SAM files containing the methylation results coming from different individuals or tissues must be processed and compared. Currently, there are two main lines for DMR identification: counting reads or number of CPGs, and counting the proportion of methylated reads by position. There are very interesting approaches from the point of view of mathematics. Almost all the tools are focused in statistical approaches like smoothing, betanomial, hidden Markov models, mixed statistical tests, entropy, but there are some tools with other approaches like logistic regression, binary segmentation, or wavelet transformation. But what about the computation performance? Actually, we developed application. The developed applications are highly computer skills dependent. Applications over platform with R package, C, C++ package, Python, Java package, scripts in Perl or R. Highly computer skills dependent, of course. Only a few tools use the GPU multi-core capabilities. By example, Bismuth or MethylKit include a user configurable parameter. GPU high parallelization is used only by HPC D-Hunter for identify DMRs and visualize methylation signals. A different aspect which reserves a special section is the use of artificial intelligence in the methylation analysis. Deep learning has been used not only for the DMR detection explained in the previous section, but used in other aspects of DNA methylation. Of course, all the proposals of using deep learning are based on the GPU, using these architectures for building the neural network required in the artificial intelligence technique. So, we can find deep learning networks that use high CPU parallelization and machine learning algorithms that use CPU parallelization. The special problem in the use of artificial intelligence for genomic analysis is the high dimensionality and low sample size of the dataset. It is well known that deep learning increases the accuracy with large dataset. However, there are solutions to make it possible. The main applications are prediction and diagnosis of diseases, mainly focused in cancer. There are predictors of single-cell methylation states with specific tools, and there are dataset generators due to this high dimensionality and low sample sets. Finally, as a general conclusion, we can state that the methylation analysis tools are often designed and implemented by biomedical researchers who do not have the necessary skills in computer system configuration or administration. Thus, they cannot use tools which exploit the parallelism in the underlying computer platform. All the aligners designed for visual field sequencing data are designed for a standalone workstation, and only a few can use as many cores as indicated by the user. Thus, they cannot exploit the parallelism in the underlying hardware platform. Most of the tools designed for DMR detection are based on statistical tools that are packages. The common feature of all these tools is that they only use the underlying platform as long as the R applications does. Therefore, we can state that they only take a limited advantage of the potential parallelism on the underlying platform. Another aspect, due to the different approaches, different tools provide different DMR sets. The validity should be determined only by the medical user. Finally, the use of deep learning becomes a new approach to methylation analysis, taking advantage of the potential of the heterogeneous hard architecture thanks to easy-to-use neural network libraries. That's all. Thanks to Mineco, Spanish MCIU program, European ERDF program, and of course of my networks and virtual environments group. Thanks for watching.